everyone. Thanks for listening to our presentation. The paper that we're talking about involves the mTOR pathway, autophagy, and ASD-like symptoms. So the reason why we decided to talk about this topic is because mTOR pathway is such a hot topic in biomedical research. It is not only involved in cancer biology, nutrient sensing, and aging, but it also affects protein homeostasis and metabolism. We are going to talk about protein homeostasis briefly here. And the reason why we both liked this paper specifically is because it is the very first paper that draws the dots between mTOR pathway and ASD, short for autism spectrum disorder. So that was just a cool thing to see, and we just wanted to read up more on that. A little bit of overview about how ASD genes impact mTOR and the mTOR pathway. Basically, the mutations in ASD genes affect abnormalities in the temporal lobe. And the temporal lobe is an area in the brain that is involved in social, it's involved in the regulation of social behavior and social connections. The ASD genes also inhibit the mTOR pathway and DISC1, DISC2, short for tuberous sclerosis. And ASD genes also cause a dysregulation in autophagy. The mTOR pathway is not, it is a very complex pathway, but for the sake of this paper, we only have to understand the relationship between it and DISC and autophagy. Basically, DISC1, DISC2 is an inhibitor of mTOR, and it acts on it by inhibiting Reb. This is supposed to be an arrow here, but Reb is an activator of mTOR, and when this is inhibited, DISC1 will inhibit mTOR. Also, mTOR and autophagy cannot be upregulated and downregulated at the same time. mTOR, whenever it's overactive, it is going to inhibit autophagy. So here the authors are proposing a general hypothesis that has been suggested by other researchers. There seems to be a connection between mTOR pathway, specifically that it is overactivated in patients who have ASD. And the overactivity of mTOR causes excessive synaptic protein synthesis. Again, we discussed here that mTOR affects protein homeostasis. So there's an increase in protein production, and this leads to the manifestation of ASD symptoms. So we just need to go over the process of neural pruning for the sake of this paper. Neural pruning is basically the loss of neurons due to low utilization. This is what happens when the neural pathways are not utilized very often. When neurons are not needed, they basically die or change connections. This is a very natural process in early development. There is approximately twice the numbers of neurons in baby brains compared to adult brains. And it also has been known for a long time that neural pruning is reduced in individuals with ASD. Neurons don't die as often, and individuals with ASD have a more flexible brain. This is not a good thing. So the hypothesis that has been specifically proposed by the researchers in this paper, it starts off with a state of overactivity in the mTOR pathway in ASD patients. And then they specify that the consequence of mTOR upregulation is that autophagy is inhibited, and synapse maturation, which is the same as neural pruning defects, it is disrupted due to the autophagy impairment. And this is just a brief delineation of the methods. The measurement of dendritic spine density is the primary method in many of the figures. It was measured by staining the Golgi using the Golgi-Kops technique, and the measurements were taken from the dendritic spines of the basal dendrites of the temporal lobe, which is a region usually implicated in ASD and communications and social behavior. For the ASD-like social behavioral testing, they include open field testing and novel object recognition, which is observing, sniffling, walking, rearing, grooming, and other social behaviors. And dialistic labeling and immunohistochemistry were done in the latest figures.
For the results, we are going to take a look at figure 1, and that is this figure here. And we are looking at the spine density. On average, we can see that they have been grouped into 2 different age groups for the control. Here we have the age group of 2 to 8, which is early childhood development. There's a higher spine density compared to that of the age group of 13 to 18 here, which shows that there is a lower spine density. And next we have the ASD, and there is a small decrease here that is being shown in spine density between the ages of 3 and 9 to the ages of 13 and 19 years old. The spine density does decrease with age, but it does not decrease as much as in the control group. And they've come up with the fact that there is less neural pruning in ASD patients. The next figure that we will take a look at is going to be figure 2 here. Figure 2 is showing that there is PSD 95 and synapsin 1, which are proteins. And here the levels of the proteins are normalized to actin. This figure here goes again by age, and starting off in the patients with the ages of 2 to 9, they have PSD 95 protein, which is higher than the ages of 13 through 20. And this is consistent with showing the lower spine pruning in the ASD patients here. There is no decrease in PSD 95 between the 2 age groups in ASD patients. This is consistent with the previous findings about the defects in synaptic pruning. In our next figure, which is figure 3H here, we are going to be looking at TIS2 plus minus models in ASD patients. When TIS2 is knocked down, mTOR is overactive. And when TIS2 plus plus, which is the wild type, represents the control group, there's normal mTOR activity. The decreased rate of spine density in the control group is higher than that of the ASD group. This confirms the fact that there's less spine pruning in the ASD group. When RAPA, which is rapamycin, is added, mTOR is inhibited. Inhibiting mTOR will bring more similarity between the wild type and the mutant.
All right, so figure 4. Okay, so figure 4 right here. It basically it's a transition to understand the relationship between autophagy and how autophagy impairment affects the manifestation of ASD-like symptoms. So here the authors used a molecule called BAFA1, and it acts by impairing autophagy. It interferes with the formation of the phagosome. Um, when we inhibit the basically when we inhibit when the autophagosome formation is defected, there is a much lower neuronal density, or which is the somatic cells density. It is greatly reduced in the DISC2 knockout group, which is the ASD group, compared to the wild type. And inhibiting mTOR is going to reduce this difference, and it's going to bring the concentration, the density of the neurons closer between the two groups. This basically means that there is a relationship between mTOR autophagy and ASD-like symptoms. And figure 5 proved this a little bit deeper. Basically, as the ATG knockout, this is the control group. That line and dotted line represents the knockout group for ATG. And ATG, if we did not explain that earlier, ATG is a protein that is involved in autophagy and the formation of the autophagosome. When ATG is impaired, there is a much slower decrease in the spine density throughout age. This means that the ATG knockout mice, the reduction in spine density in the knockout group may result from an increase in synaptic formation. So this last figure, it shows that basically the authors were trying to investigate and eliminate other potential experimental errors that can arise. Basically, the reduction in spine density can be either a result of a decrease in neural formation the increase in neural formation or a decrease in neural elimination or spinal pruning. So they knocked out the ATG protein through RNA editing, and they realized that there is no impact on neural formation when autophagy is impaired, but autophagy only affects the neural pruning, which further proves their hypothesis that it is a probable valid hypothesis. And in conclusion, the authors reiterate that mTOR regulated autophagy is required for developmental spine pruning and activation of neural autophagy, correct synaptic pathology, and social behavior deficits in ASD models with overactive mTOR.